gentlemen. Hello to everybody. Boy, it's a hard crowd to quiet down this morning, but we'd like to begin. And I first want to just run through a few things. We have a number of people who are not actually residents here. First and foremost, we have a share of our Academy family from Uni Hill here. So a very warm welcome to all of them and all of you who are residents here, and of course to all our residents. We're super excited about today. We have a couple of ground rules. One is if you need to use the restroom, it's right out the hall and on your right. There's water and coffee over here. Please silence or turn off those boxes that you carry around. That would be really helpful. We love that. We will have time for a Q&A afterwards. And when you get what we try to do is get a microphone to you, because it's much better for everyone to be able to hear. When you get the microphone, model this. It's a little bit like, I don't know, Lena Horne or something. You got to do the singer style and hold it about 3 or 4 inches from your mouth, and then we'll hear you very well. So thank you for that. And afterwards, after the Q&A, Tom has offered to sign his book. So anybody who has his book, this book right here, The Catalyst, will be over at this table afterwards. So come join Tom there. I got the cell phones. I think that's it. I want to turn it over now to Miss Jeannie Thompson, who has actually been the force behind this. She got this all to happen, and she's going to introduce Tom. And so huge thanks to you.
Oh dear. Here we go. Thank you. Am I good? Okay. First of all, I do have the prop, the book. Okay, can you hear me? Okay. It's our pleasure to have Tom Cech here with us today. He is the Distinguished Professor of Biochemistry here at CU and an investigator with the HHMI Howard Hughes Medical Institute in Chevy Chase, Maryland. After getting his PhD in chemistry at Cal Berkeley, he became a young professor and researcher here at Boulder. During the early 80s, he and his lab discovered the importance of RNA. It's much more than a passive, transient information carrier, as was earlier believed. In fact, it was the catalyst. In 1989, he became CU's first Nobel laureate, and he remains the only one in chemistry. The other 4 are all in physics. I've heard he no longer keeps his metal under his mattress. The metal made it too lumpy. The metal made it too lumpy. In 2000, he left Boulder for almost a decade to become the president of HHMI. Fortunately for us, he returned to become executive director of the new BioFrontiers Institute that was being established here at Boulder. It's been in place for 15 years now, and Jack and I are pleased to have become involved with this institute. Several years ago, Tom stepped down as the executive director to focus on research and teaching. He is considered one of the best teachers here at CU, and he loves teaching undergraduates. He's currently the faculty director of the Institute's interdisciplinary quantitative IQ biology PhD program. Besides the Nobel, Tom has received numerous awards and prizes, among them the National Medal of Science. He's also been elected to memberships in the National Academy of Sciences and the National Academy of Medicine. If you look at Tom's Vita, CV, it's 50 pages long, and 42 pages, I looked at it, 42 pages are lists of publications. It's amazing. He's founded companies and served on corporate scientific boards. He's a devoted Coloradan. He and his wife, Carol, enjoy everything about the state, and we're thrilled that he's here with us today. Tom.
Well, thank you, Jeannie. That was a very personal introduction. Really appreciate it. I think I should turn on my microphone. Just saying. Hello, hello. There we go. Looking in the around in the audience, I see so many friends. I wonder why I haven't moved here. But so today I'm going skiing this afternoon. So I need to get on with the show here. And what I'd like to talk about is this book that I wrote, that Jeannie, you better take a copy or I'll steal it, The Catalyst, and about my pathway to writing this book. So it really sort of started during the pandemic, both because I was at home and had some extra time, but also I was persuaded by data like this. This is the fully vaccination rate for COVID-19 at the last month when international data were still being kept, which is about 1.5 years ago. And you can see we didn't do all that well. You can also see that a number of countries, which when you visit them, don't seem that much different from the US. For example, Australia and Canada had substantially higher rates of full vaccination. And I realized that the reason was because, Americans don't like to be told what to do. They don't like to be shamed into doing something. They sort of often do the opposite if someone tells them what they have to do. So that maybe telling people that if you're a good person, you better get a shot was not all that successful. And that instead, if they perhaps just had more information, more confidence in the technology, without being told that they should get vaccinated, that they might make their own decision. And by the way, I don't know for sure that that's a fact, but it seemed reasonable to me, and so I decided to write a book, not just about the vaccine, but about this wondrous molecule, RNA. And I thought this would be easy for me because I've taught more than 2,000 freshmen in general chemistry at CU Boulder. And I thought I was rather good at reducing complex scientific topics to an understandable level. But it turned out I was wrong, because college freshmen have just finished having a lot of science in high school. They've often had advanced placement courses, whereas their parents and their grandparents haven't had a science course in decades. And when they did have a science course, RNA was probably not a big part of it. And so the audience really is not equivalent to college freshmen. It's equivalent to you guys, and who are bright and inquisitive, but are not people who've been studying chemistry or biology recently. And so my editor, Jessica Yao at Norton, whenever I send in a chapter, she would say, "It's too technical." I said, "What? I worked for days on this to try to simplify it." "No, it's too technical, back to the drawing board." And so she was very helpful in this. Now, why is it so hard for scientists to talk to the general public? There are multiple reasons, but I think a main one is this jargon, which is not a bad thing. I mean, it really facilitates our conversations within the discipline. It doesn't have to be even within, biochemistry. It could be within investment planning or something like that. People have their jargon, which allows them to say something that would take a long time to explain in just a few words. I can walk into the laboratory and say, maybe you should just CRISPR that allele into the endogenous locus in HEC293 T cells. And the student pretty much gets what I'm saying from that sentence. It would take me 3 days to explain that to the people here in the audience, right? So jargon is not intrinsically a bad thing, but we have to learn as scientists that it's an impediment when we're talking to a non-scientific audience, and we have to quit using it. And that's what I tried to do in my book, The Catalyst, and it's also what I'm going to try to do today. I'm not going to talk about the book per se. I'm just going to give a few highlights of the topics that were covered, some of the topics that are covered in the book. And I'm going to try really hard to avoid jargon, to use simple, understandable language, and at the end you can walk up to me privately and tell me where I failed to do that, okay? So the topic of ribonucleic acid is an exciting topic, not just because it's what I work on, so therefore easy for me to talk about, but I really believe that this century is the age of RNA. DNA is past stuff. That's last century stuff. We understand how DNA works, and its interest is actually rather limited, right? It just encodes information. And it's only when that information gets copied into RNA and sometimes ultimately into proteins that the action occurs, right? So really this is a topic that I think everybody should know about. And it's difficult because for DNA, everybody's already very comfortable with it, right? They know that many of their traits are inherited through their DNA, including both positive traits but also propensity to various inherited diseases. And they're concerned about that because they want to know whether a particular disease gene is running in the family. They know that they can trace their family history, do genealogy through DNA. They know that crimes can be solved through DNA evidence. But somehow when you come to DNA's daughter molecule, RNA, ribonucleic acid, people say, oh, that's much too complicated. But it really carries the same information as the DNA double helix in a single strand instead of a double helix. In fact, if you would read out the bits of information along one strand of the DNA, it would be some order of the 4 bits of information, A, G, C, and T. Then in the RNA, you would see exactly that same information copied verbatim. A, G, C, and U, U being a slightly different chemical modification of the T in DNA, but it has the same informational content and the same ability to code. So what makes it a little bit easier now to talk about RNA is that most people have at least heard about one kind of RNA, messenger RNA, abbreviated mRNA. And they know that the SARS-CoV-2 vaccine that was produced both by Moderna and by BioNTech in Germany had mRNA packaged up in a grease ball called a lipid. Lipid is a technical term for a greasy molecule, nanoparticle, very small particle, which is able to then produce a protein when it gets into our body. The mRNA is decoded, it's read out to produce a protein that looks like the envelope protein, the spike protein, on the outside of the SARS-CoV-2 virus. And this gives our immune system a heads up. It says, be on the lookout. If you see something that looks like this, it's bad news. You should be making antibodies against it. You should be killing any cells that are infected with that particular virus. And by the way, the SARS-CoV-2 virus is itself an RNA virus, which shows you how sort of marginally important DNA is. The virus has all of its information encoded in RNA, doesn't use DNA at all, and it makes all the proteins it needs by reading out its own RNA. So this is a battle, an epic battle then, of RNA vaccine against an RNA virus. Now, when I think about why people were suspicious of the mRNA vaccine, and they continue to be, we hear about this in the news, like this morning, right? There are people who don't believe in vaccines. And I think one of the reasons is that many magazine articles, books, social media talk about mRNA as a medicine, mRNA as a drug. And that makes people nervous, it makes them suspicious. And I think instead we should emphasize that mRNA is natural. It's in every cell in our body and in every other living organism on the planet. It's in all of the food we eat, whether you're a meat eater or a vegetarian, you're eating mRNA all the time because it's in all of living things. In fact, in Boulder, we might even say it's organic. So not only is it natural, but we know how it works. We've known for 60 years. This is not some newfangled invention. We know that the DNA code is copied, the information is copied into RNA, and then groups of 3 of these bits of information at a time are deciphered to encode a particular amino acid, be it lysine or tryptophan or phenylalanine, that are lined up in a protein, and the proteins are the main movers and shakers in all of our cells, right? They are the proteins that allow our muscles to move and make our limbs work. They're the enzymes that are digesting the breakfast that you have in your stomach. These are all protein molecules. And each of those proteins has a messenger RNA that encodes it, and that messenger RNA was copied from a gene made of DNA. Unless it's a virus, then just it's directly copied from the RNA without the DNA. Now, what the folks at the vaccine making world could have done would have been to start out for the gene for this SARS-CoV-2 spike protein and copy that into messenger RNA and copy that into protein to use as a vaccine. But what they realized was that they could take a shortcut. They didn't need the DNA. They could just inject the messenger RNA and let the human body do the heavy lifting of what we call translating that mRNA into the protein. And that's how these 2 vaccines were in fact made. Now, the other thing that people complain about with respect to the vaccine is that it wasn't perfect. And that's absolutely true. And here are a few of its imperfections. So although it did protect against hospitalization and death, reinfection certainly occurs with vaccinated people. That's a property of the virus, not a fault of the vaccine. Many respiratory viruses have this reinfection problem. But, you know, protecting against death, that's a pretty big thing. And in fact, it's hard to estimate exactly how many lives were saved, but somewhere between 10 and 20 million people didn't die during the first year that the mRNA vaccines were rolled out. So when your neighbor or your, science suspicious friend says, the vaccine isn't that good, say, well, it isn't perfect, but it does save hospitalization and it saves a lot of lives. When the virus mutates, then there's immune escape and the vaccine isn't as effective anymore. And that's, of course, a shortcoming. And the other thing, of special concern when we try to roll this out to third world countries, is the fact that it has to be stored and transported at ultra cold temperatures. And that's something we need to work on, right? Because that's a shortcoming, and I think that will be solved with a little bit more time. But nonetheless, there are some huge advantages of these mRNA vaccines. So it's so quick and straightforward to retool it to match the new genetic information in a mutant virus that you can adapt it to respond to variant viruses rather quickly. And you've seen that over the last couple of years. And the other thing is that, do you know how your flu vaccine gets made? It gets made by hand injection of 1 million chicken eggs per year in the United States with a debilitated form of the flu virus that cannot give you an infection. That takes not only a lot of omelets are wasted there, but in addition, it's very slow. So they have to start making next year's flu vaccine by guessing what strains of flu will be rampant during the next flu season. And their guesses aren't all that great. Typically, the flu vaccine is between 30 and 60% effective. unlike the measles vaccine, which is 99+% effective, for example. And so that's something that the mRNA vaccines have a real advantage because they can be designed and produced so quickly that we can wait until we know what flu is going to be around in the next flu season and then start making the vaccine. And that's in progress now, and we're hopeful that within the next few years there will be more effective flu vaccines tailored to each year's variant of the vaccine. Personalized cancer vaccines are in clinical trials now, and they're looking promising, but I can't promise that that will happen. But this is a very exciting area for mRNA in the future. And then even more speculative, but also exciting, is the possibility of therapeutic mRNA. There are some 3,000 diseases that are fairly rare that are caused by the failure of our body to make a particular protein because of a genetic mutation. You know the names of some of these: muscular dystrophy, cystic fibrosis, sickle cell anemia. And if we could put the correct protein back into those people, that would be a therapeutic advantage. And so can therapeutic mRNA be used for this? It'll probably in some cases yes, in some cases no. Can we make enough of the protein and get it in the right part of the body to be therapeutic? Each of these is going to be, quite a bit of research is going to have to be done. So, in any case, if RNA could only be a message, even that might qualify it for this being the age of RNA, because that's pretty cool, to being able to encode all of the diverse proteins that are present in all of the organisms on the planet. But I've got a little time left, I see, and I've only done mRNA. Well, maybe there's something else that we could talk about. And this is the world of non-coding RNAs. These are RNAs that don't care about making protein at all. They function at the level of the RNA itself. And there are a dozen or more major categories of these non-coding RNAs. There are hundreds of thousands of examples. And because of the time constraint here, I've decided to talk just briefly about 3 of these non-coding RNAs, ones that I have some connection with and we work with in our own laboratory. The 3 kinds of non-coding RNAs are catalytic RNA, the ability of RNA to speed up a reaction that's essential for life. We call these ribozymes, ribo meaning ribonucleic acid or RNA, and zyme meaning that it has some enzyme-like activity. Secondly, RNA can bestow immortality. Wow, that's a biggie. But before you get too excited, only at the level of individual cells, not at the level of the whole organism. Okay? And then finally, RNA can get its sort of ultimate comeuppance on its parent DNA. It can actually cause the editing of the DNA and change the code in the DNA. And this is RNA-powered CRISPR gene editing. So the first of these, the ribozymes, actually is my own story, and it starts when I moved and set up my laboratory here in Boulder, and the first thing we started working on was, of course, pond scum. Wait a minute, why would you want to work on pond scum? Well, it wasn't just any old pond scum, it was this organism, Tetrahymena thermophila, a single-celled animal that inhabits freshwater ponds, Varsity Pond on the Boulder campus, all the way to ponds on the other side of the world. And it was intriguing to me because it had 10,000 copies of a particular gene. And if you're a biochemist, you sort of like a lot of stuff to work with. And 10,000 copies per cell is a lot. Compare that to a human cell. how many copies do you have of each of your genes? You know this, right?
Yeah.
How many? 2, right? One from mom and one from dad. So 10,000 is a big deal. And what we found out, surprisingly rather early, was that these 10,000 genes were all interrupted by a stretch of non-informational DNA called an intron. You can think of this as a commercial message interrupting your favorite film or TV show, right? You grab the remote and you fast forward through it. Well, the cell doesn't have a way of fast forwarding through these introns, and in fact they are copied along with the useful flanking sequences, which are shown in blue and green, as part of a large RNA, which then has to be cut and has to be joined together in order to give the functional RNA. So you have to take that commercial message, splice it out, and rejoin the code to be useful. And it turns out that this had been discovered just 2 years before I came to Boulder. We didn't discover it, it was discovered at MIT and at Cold Spring Harbor Laboratory on Long Island in New York. But already it was known that most human genes were interrupted not just by 1, but typically by 5 or 10 of these introns. And so this splicing out of this sort of misinformation is something that is critical to human life. But how did it occur? What was the mechanism? And here we had a gene that was present in 10,000 copies, and so this process was happening in spades, and we thought maybe we can be the ones to figure out the mechanism of this RNA splicing. Well, we ran into trouble because the splicing worked so efficiently every time we isolated the RNA that we were confused about what the catalytic power that was making the splicing work, what that might be. And we knew there had to be a protein enzyme that was cutting and pasting the RNA together because it was happening with such incredible facility, such high speed, such great accuracy. And ever since James Sumner from Cornell University won the Nobel Prize in Chemistry some 30 years before we were doing our experiments, he proclaimed that all enzymes are proteins, and in those 30 years in between, every enzyme that had been found was a protein, so people quit worrying about this, right? Enzymes are proteins. Now we had an RNA that was splicing without us adding any proteins. We thought, well, maybe it's contaminated by some protein enzyme that came along for the ride, and so that was a possibility, and my graduate student, Paula Grabowski, presented me with this at a departmental Christmas party. You know, is there a hidden protein in our preparations or not? If not, then that means that the RNA by itself is the biocatalyst. And we were able to prove that hypothesis that the RNA was catalytic, and this led to then the state of Colorado's first Nobel Prize in 1989. You can see I'm a bit younger there. My daughter Allison was allowed to come and dance at the party. My younger daughter Jennifer, who now lives in Seattle, was still a baby and she wasn't allowed to come to the ceremonies. So I could talk more about this and I could talk for hours about it, but instead I'm going to get on to immortality and tell you just a little bit about telomerase. Now, in order to define this technical term, telomerase, I have to first tell you what a telomere is. And this is a Greek word. Telos means and as far as I can tell, mere means stuff at the end. So this is the stuff at the end of what? Of each and every one of our chromosomes. And you can see a picture here of the 23 times 2 human chromosomes that are present in every what we call diploid cell in our body. And what we've highlighted on this picture with the yellow dots is the DNA that's at these telomeres, at the very ends of the chromosomes. And you can think of this telomere as sort of a protective cap at the ends of our chromosomes, just like the plastic tips on our shoelaces that prevent them from unraveling. What are those plastic tips called, by the way?
Aglets.
Aglets, thank you. You must be a crossword puzzler. We're a crossword puzzler. Okay, so what happens to these tips at the ends of our chromosomes is that as the cells divide, if you don't have some active mechanism of maintaining those tips, then the DNA shrinks from the ends of the chromosomes. And you may think that's a bad thing, but it's sort of a protective mechanism. It allows the cells to divide sort of like a clock, right? Saying, okay, you've divided enough, it's time for you to stop dividing. Because once you're a mature adult, you don't want all the cells in your body to continue to grow. They have to be regulated and they have to stop growing. But how about in certain cells that do need to continue to grow, like stem cells that are needed to replenish the aging cells in our bodies? Well, those need to maintain their telomeres, and that's where this enzyme telomerase comes in. And it is this wonderful collaboration between a protein subunit called telomerase reverse transcriptase, again, sorry for the technical name, this yellow egg, which was discovered here in Boulder in my laboratory, and an RNA molecule, which was discovered by Liz Blackburn and Carol Greider, who got the Nobel Prize for the discovery of the telomerase itself, including the RNA part. And what the RNA does is it provides a template sequence that allows the enzyme to add these units to the ends of our chromosomes, and wherever there's an A in this template, a T is added. Wherever there's a C, a G is added to the DNA at the ends of our chromosomes. So by counteracting that telomere shrinking, this gives a cell immortality. Well, immortality is sort of very attractive to a lot of people, and so it's perhaps not surprising that if you go on Amazon.com, you will find dozens of products that are, well, I don't know if they're guaranteed, but they are said to elongate your telomeres. See, clinically proven to lengthen telomeres through telomerase activation. And I think they might have some activity, hopefully not too much activity, because there's one other kind of cell that needs to activate telomerase in order to be immortal, and that is called a cancer cell. And it was this guy, Levi Garraway, who, when he was at Harvard Medical School, and his med fellow, Franklin Wong, who's a friend of mine, discovered how tumors activate telomerase to gain immortality. And it was a mutation in the control region of this gene that we discovered here in Boulder, the gene for the protein subunit of telomerase. And quite remarkably, that mutation occurs spontaneously 300,000 times per year around the world and gives rise to melanoma. So if you take melanoma tumors from diverse patients in Sweden and in Denmark and in Russia and in South America and in Boulder, you will find that 80% of those have activated the telomerase gene by this mutation discovered by Garraway and Wong. It's also very common in brain cancer, glioblastoma, bladder cancer, liver cancer. Interestingly, it's absent in other common cancers. They still have to reactivate telomerase, but they do it by a mechanism that is still not really very, very well understood and does not involve this particular mutation that I just talked about. So again, a double-edged sword. On the one hand, longer telomeres are good for longevity of cells. On the other hand, cancer is a possible outcome if telomerase is upregulated. And then finally, I'll just say a few words about the fact that RNA, non-coding RNA again, not a message, can guide the editing of our genome. Again, sorry for the technical term, but the genome is the total constellation of genes that we have in every cell in our body. So it's that whole ensemble is called the genome. And this is called CRISPR gene editing. So how does this genome editing work? And again, this is what I do in the Catalyst over and over again, is to try to have sort of everyday analogies, metaphors that will help people understand these technical concepts. So genome editing is like text editing. It's like find and replace on your laptop software. So for example, if you have a typographical error in the middle of the text, you can edit that and replace it, the U for an A. But it's a daunting problem because the human genome in a typical font size would take up about 1 million pages of a book, single space typed, okay? So this CRISPR genome editing is so remarkable that it can search through all of that text, 1 million pages, and find the one typographical error, and then catalyze its conversion to the correct sequence. And we do this in the laboratory. It's so facile that undergraduates in my lab have successfully employed CRISPR genome editing. Not in people, right? In individual cells growing in petri dishes. It's not considered ethical for us to do experiments on human subjects with this work. Now, this was so exciting that it led to the 2020 Nobel Prize in Chemistry to Emmanuel Charpentier from the University of Vienna and Jennifer Doudna from UC Berkeley. And we were particularly excited about this in Boulder because Jennifer was one of my postdoctoral fellows. In fact, here she is with some of our other faculty around the country who trained in Boulder. 56 of my trainees have become professors, and the ones shown here are professors at, in addition to Berkeley, University of Chicago, University of Washington in Seattle, and the Scripps Institute in La Jolla, California. Now, how does this CRISPR genome editing work? Well, it's a bit like this, but this is a little oversimplified. Okay? It's not just the red scissors. It sets up a cascade of events, of repair events on the DNA that end up being able to program in a change of one of those letters of the DNA for another one to correct an inherited disease or just a spontaneous mutation in a disease. So because of all of these great advances that we found about non-coding RNAs, I think it justifies the conclusion that the 21st century is the age of RNA, and there are many more of these that remain to be discovered or that have been discovered, and their function, their role in the body is still being debated. And so with that, I would like to thank you for your attention and entertain questions.
So it's time for you to close your desks, put your books away, and now Kevin's going to help me hand out the test. You didn't go for that. Kevin has a mic and I have a mic, so anybody have a question?
Remember, you have to hold it close up for it to work.
Anybody?
It can be a question about anything. It doesn't have to be about what I talk about.
So I was just thinking, couldn't you just call the vaccine an organic vaccine?
Yeah, right. That might help. You know, it does have it does come from a GMO synthetic gene. That's the way we make the RNA, right? So the RNA that goes into the vaccine, we do make it from recombinant DNA. So we probably couldn't really call it organic.
This is great, and I actually understood probably 80 to 90% of what you said. However, the question really is...
My neighbor said 10%, by the way, so thank you very much for that.
The question really is, given what you're laying out there, how close are we to really solving cancer as a disease using this technology?
Well, you know, I have to be careful what I say, because many of us have lost and continue to lose loved ones through cancer. But the truth is that it is not the disease of the future, because cancer rates have been going down rather steadily for the last 20 years. And that was before this new technology called immuno-oncology came out, which is saving lives, giving durable cures to patients who would have been dead a dozen years ago. And so, although cancer remains a challenge, it is much more under control than neurodegenerative disease. And it's really, that's the area that modern medicine has made the least impact on, and it does not even appear to be on the verge of making an impact on Parkinson's disease, on ALS, on Alzheimer's disease. So when I talk to various charitable foundations that are interested in putting even more money into cancer research, I say, you know, we're working on that, it's important, but I think there's this other unmet opportunity that's even more important. And RNA will play a role there as well.
Did Tom have a question?
No, Tom, no. The next question's over there.
So, as a young person, what kinds of things influence you to be curious and to consider work in this field?
Well, I knew I was going to be a scientist from the time I was in first grade. And and it was geology that was the science that I was mostly interested in. I had a rock hammer. I would go around the neighborhood cracking open every rock that I would see to see what was inside. And my parents were very supportive of my interest in science, which is also not true for everybody, but it helped me a great deal. And as time went on, my interest evolved into astronomy, and then into physics. And finally, when I failed the entrance exam for chemistry in college, I said, damn it, I need to know this chemistry stuff. So then I became a chemistry major, and I was never interested in biology. That was too soft of a science, wasn't quantitative enough. And it was only in graduate school, when I went to graduate school in chemistry, that I realized that although I loved learning about chemistry, I didn't really like doing the experiments with my own hands. And that's when I switched over to biochemistry and just loved it. And in the meantime, biology had become much more of a rigorous science as well, because we started to understand how living systems work at the level of atoms and molecules, not just at the level of the organism, which is obviously also an important level to understand. So that was my pathway.
Hi. So I'm really intrigued with the 21st century age of RNA. It seems like it's really more about an age of abundance, and that this is an important part of the story, but I think it's more than a coincidence that renewable energy promises energy independence, which may end autocratic supply, and that AI is helping us expand our cognitive capacity to the extent that we may be able to change our minds once in a while. So,
Absolutely.
Is this all part of something huge that's happening?
Well, yes. It is definitely, and you're absolutely right. I mean, I'm pitching, RNA because I'm an RNA scientist, and it's not to at all belittle these other critical areas. And I think that, in fact, the most critical area which is now being quite disparaged, is climate change. Because if we don't have a planet to live on, RNA doesn't make any difference in the long run, right? So I appreciate your comment, thank you.
I'm curious what the life expectancy is here in America compared with, say, 10 years ago, and how that is impacted by your research, and how the life expectancy here in the US compares with that in, say, Europe or other parts of the world.
Yes, it's flattened off to a great extent, the life expectancy, although the distribution of old versus young people is becoming increasingly skewed towards older and older folks. You guys in the audience are very soon going to be the average age of the American population. You know, that's just that's the way it is, and I think modern medicine has helped us many people live a healthy life until an older age. But what's happened is, as the cancer death rates have gone down, the neurodegenerative death rates, which scale a lot with age, have gone up, and so it's sort of compensated. Japan is an interesting example of a country that has considerably longer life expectancy and healthier older population than the US. And it's hard to know quite why. It could, there are probably multiple reasons. Diet maybe is one. I think they may be, there's less of an obesity problem there. There's maybe overall better diet. But in addition, they spend half of their healthcare dollar on diagnosis, on checkups, on health, rather than on disease. You know, and that, I think, is a healthy thing to do. And this country, it's all about popping pills when you're sick, and not so much about a healthy lifestyle in our medical care. So maybe that's something that I know. I just signed a letter not to confirm RFK, but nonetheless, I will say, if he could pull that off, that would be a wonderful thing. This, you know, a sort of a energizing interest in diet and health.
So what is the life expectancy here in America today compared with 10 years ago, roughly?
Oh, I think I think it's gone up only just a little bit, and
Can you name a figure?
yeah, if I do, I mean, somebody can Google this while I'm talking. I don't know.
You're talking the oldies but goodies.
It's around 80. I don't know what the average is. Yes.
I'm recalling the moon, you know, the moon shot.
Yeah.
And it seems like, if I recall my history correctly, when Kennedy was arguing that we need to go to the moon and bring people back safely, he was trying to convince Congress for funding. And his advisors said, you don't have, your funding problem is a marketing problem. You need a public relations effort to convince the country that this is a good idea. Well, okay, there you have the president, in this case, NASA, and so on. What is the entity that can solve the social marketing question or problem that you mentioned right at the beginning?
It's really challenging, because I think what's happened, you know, as all of you know, over the last 20 years, is that social media have taken over for young people their main source of both information and disinformation. And whatever you hear and you know wonder if it's correct, you can always find somebody on the internet who will agree with any theory, no matter how little evidence there might be to back it. You know, a good example is vaccines cause autism, which was based on fraudulent research that was completely made up and was removed from the scientific literature, but the idea continue to percolate on the internet. And if anybody wonders whether this is true, you know, vaccines cause autism, just Google it and you'll find lots of support for that theory, even though there is no scientific basis for it at all. And it's been completely debunked. And so that's an example, right?
It seems like the alternative, since there is no national effort at social marketing, the only ones doing it right now are the pharmaceuticals, which is like the kiss of death right now. Or, I shouldn't have said that, I guess.
Right, well, it seems so self-serving for them to be advocating for these things because they're making a profit, right? So it has to be outside of the pharmaceutical realm. And, you know, I'm involved in multiple agencies which are trying to get better communication. But one thing that when I go around and give talks like this, I gave 7 of them at different universities and in Canada as well as the US in the fall, I always tell the students in the audience that it's your responsibility to start, you know, communicating your own science. Why are you doing what you're doing in graduate school, right? And why does it make a difference in the world? Get rid of the jargon, take some training in science communication, and go out and talk to your family, your neighbors, your church group, your sports group, go to a grade school and talk to an elementary school, you know. So I think we need to activate scientists to be more part of this solution. It may not be enough, but it's one way we can help.
I was curious about one thing. I saw a statistic yesterday at the Museum of Boulder that said that Boulder has the lowest percentage of vaccination rates. And it didn't say what kinds of vaccinations, so I don't know. I wondered, first of all, how you would explain that in this kind of educated community. And second, why did you put the medal under your bed in the first place?
Well, for the first question, I don't know that I have an explanation. We have Boulder itself ties with Cambridge, Massachusetts as having the highest percentage of the citizenry having a post-baccalaureate degree of some sort. So clearly, this is an extremely educated population. On the other hand, the National Anti-Vax Center is headquartered in, is it Lafayette or Louisville? So I don't, you know, a lot of free-thinking people, and they think all kinds of different things. It doesn't seem to scale just with the amount of education you've had. And in terms of the medal, that was just a joke.
I have a question.
We hear so much today about artificial intelligence. So the question's really simple.
I'll repeat it.
Does artificial intelligence have any promise for what happens in labs such as yours?
Okay, so the question is, what is the impact or the possibility of impact of AI on the research that we do? And the answer is it's already having an impact. In fact, the way in which these large biological molecules fold up in three-dimensional space is a key to understanding how they work and also to targeting them with drugs that might inhibit their action. This used to take many years and be a real hit or miss proposition. And now with AI, we can predict the folding of both large RNAs and large proteins with increasing accuracy in 30 minutes on the computer instead of 2 years in the laboratory. In fact, we had a student who spent 2 years solving a structure of a molecule, and then AlphaFold 3 came out, and in 30 minutes she got a structure that looked really very similar to what she had spent 2 years working on. And so that's both, you know, discouraging and encouraging. Discouraging for the student, but encouraging for the field.
Going back to
RNA and mutations, it seems that you can identify the mutations, but is there any understanding of the causes of the mutations?
Oh, yes.
And then I would like to add about getting the message out. It seems that maybe teaching graduate students how to be influencers on social media might help.
Exactly. Because people like me and many of you are probably not all that active on the social media platforms, but the young people are, and that's what they listen to. But your first question, remind me of the first question.
Oh, so the first question is, we can identify mutations, but do we understand the cause of mutations?
Yeah, so some are inherited. Those are called germline mutations because they come in through either the sperm or the egg DNA from your parents. But in addition to those, there are several other causes of mutations that are not inherited. And one of those is environmental insults, cigarette smoke, and that's mutations that cause lung cancer. Ultraviolet light is directly absorbed by DNA and can lead to mutations, and that's what gives rise to melanoma. Too much, not enough sunscreen, too much sunlight, and other environmental effects. And then on top of that, there's just the fact that the copying machine that copies the DNA in our chromosomes, although just fantastically accurate, is not perfect, and it occasionally makes a mistake. And so we call those spontaneous mutations, and those would be hard. That would be the one category that would be really hard to get rid of because it's already at a low level. But if it happens in the wrong gene at the wrong time, it can have severe health consequences.
Any dietary origins?
Well, yes, there are chemicals that are found in foods that can be mutagenic as well. I think maybe the greatest health danger, though, in some of the food that we eat is more the obesity epidemic than it is and diabetes than it is probably the mutations.
But any opinions about what we're hearing now about ultra-processed food?
Yeah, again, I don't think those, I think those should be minimized for good health, but I do not think that mutations are the main reason we should avoid them.
Do you have time for one more question?
Sure.
And I want to first read to you that the CDC estimates life expectancy at birth in the US increased to 77.5 years. This is in 2022, up 1.1 years from 76.4 in 21, but still down from 78.8 in 2019. The average life expectancy at birth among comparable countries was 82.2 in 2022.
So I just turned 77, so I guess I have another half a year. Thank you very much for your questions.
If anybody has a book they'd like to have signed, come on over here, and then we'll let Tom go skiing.